Good morning to one and all present here. On behalf of Seva Sadan's R.K.Talreja College, Ulhasnagar and the Microbiology Society India, I, Dr. Parimita Sharma, extend a heartfelt welcome to our respected principal, distinguished dignitaries, esteemed resource persons, faculty colleagues, research scholars, and enthusiastic participants joining us from across the globe for this 2 day international conference on AI and in silico insights in biological sciences. We are gathered here in the true spirit of inquiry and innovation. As Stephen Hawking once remarked, intelligence is the ability to change. Artificial intelligence and in silico approaches embody this very power of change, transforming biology from observation to prediction, from experiment to stimulation, and from understanding life to reimagining and re engineering it. The impact is evident at the highest level. The 2024 Nobel Prize awarded to David Baker, Demis Hassabis, and John Jumper recognize groundbreaking achievements from creating new proteins to using AI to solve the long standing mystery of protein structure prediction. These pioneering milestones not only celebrate human ingenuity but also capture the very essence of our conference, where AI meets biology to unlock new horizons. Louis Pasteur wisely observed, science knows no country because knowledge belongs to humanity and is the torch which illuminates the world. In that same spirit, this conference becomes a torch, bringing together brilliant minds, fostering dialogues, and igniting collaborations that transcribe or transcend boundaries for the advancement of science and society. Every great beginning seeks the grace of wisdom. Let us now invoke the blessings of Ma Saraswati, the eternal symbol of knowledge, creativity, and learning through the Saraswati Vandana. I request everyone to join their hands and immerse in the Saraswati Vandana.
या कुन्देन्दुतुषारहारधवला या शुभ्रवस्त्रावृता या वीणावरदण्डमण्डितकरा या श्वेतपद्मासना या ब्रह्माच्युतशङ्कर प्रभृतिभिः देवैः सदा वन्दिता सा मां पातु सरस्वती भगवती निःशेषजाड्यापहा शुक्लां ब्रह्मविचारसारपरमाम् आद्यां जगद्व्यापिनीं वीणापुस्तकधारिणीमभयदां जाड्यान्धकारापहा हस्ते स्फाटिकमालिकां विदधतीं पद्मासने संस्थिता वन्दे तां परमेश्वरीं भगवतीं बुद्धिप्रदां शारदा
Allow me also to share a few words about the proud institution hosting this academic endeavor. Seva Sadan's R.K.Talreja College of Arts, Science and Commerce, established in 1961 and affiliated with the University of Mumbai, stands tall with its vision of education for empowerment. Over the years, it has blossomed into a premier center of learning, offering diverse undergraduate and postgraduate programs powered by dedicated faculty and well equipped laboratories. We take immense pride in being recognized by the government of Maharashtra as an Acharya Chanakya Kaushal Vikas Kendra training center, a recognition of our commitment to skill based education and nation building. Today, R.K. Talreja College is not just a place of learning, but a hub of ideas, collaborations, and opportunities inspiring students, researchers, and educators alike. With this proud legacy, it is only fitting that our college becomes the stage for this international dialogue where AI meets biology to script the future. It is often said great leaders don't just light the path, they ignite the flame of inspiration in others. We are truly blessed to have such a guiding light in our respected principal, Dr. Professor Dinesh Kala sir. With over 35 years of teaching and research experience, Sir has mentored 4 PhD scholars, guided 10 major research projects, and continues to pioneer impactful work in earthquake sensors and renewable energy. Under UGC funding, he established a unique green lab, authored more than 10 books in physics and electronics, and delivered numerous keynote talks and guest lectures on national and international platforms. As a BOS member at leading universities, his academic and leadership contributions remains truly remarkable. With immense pride and honor, I now invite our respected principal, Dr. Dinesh Kala sir, to deliver the welcome address and officially inaugurate this 2 day international conference on AI and in silico insights in biology.
Good morning, everybody. You're hearing her,
Good
Principal
morning, sir.
College, Professor Dinesh Kala. I'm happy to see that the biological component of our college, that is the biological sciences, has taken a very important and interesting topic on this particular international seminar or international conference with a nice introduction by Parmita Madam about the college, about me, and with the blessing of Ma Saraswati, I'm sure this program will go very nicely. Everybody will learn. This intellectual interaction worldwide is going to be very important for every learner and person who's presenting the papers and making people aware of what exactly is going on in the present scenario. Wish you all. See, we are online, so there will be many interruptions. There may be many glitches. As a principal of the college, it's an you have to forgive us in the beginning itself. Please coordinate and cooperate. Whatever, wherever you can participate in the best possible way, and make the program successful. Thanks to Meena ma'am, thanks to Ranjana, thanks to Pramita ma'am, thanks to Jitendra sir, thanks to Ganesh sir, and thanks to Kuldeep sir. The team is very strong. The molecule is non-breakable. Thank you. God bless you all. As a result, the final is the program inaugurated now. Thank you.
Thank you, sir. Now I call upon everyone to switch on their videos for a group photo. Kindly switch on your videos for a group photo.
Ramita, announce again. I think they're not understood.
I request everyone to switch on their videos for a group photo.
Wait. I'm taking it.
Thank you all. Thank you very much,
ma'am.
sir.
Wait, ma'am.
Wait. Okay.
You permit everybody to have the video and video on for the time
Video
being.
on.
Yeah, you have to give the permission.
Okay.
That's why people are not able to come.
Last one.
Okay, should I continue?
Last one.
Okay, last one.
Photograph is yet to come.
Yeah, done, ma'am.
Okay, thank you. Thank you very much, sir, for your inspiring words and for officially inaugurating this international conference. Now,
Thank you.
moving ahead with the proceedings, it is my privilege to invite the convener of this international conference, Dr. Ranjana Khade Madam, the guiding spirit behind this event. With her vision and dedication, the theme AI and in silico insights in biological sciences has taken shape. I now request Dr. Khade Madam to kindly share her thoughts on the theme of the conference.
Yeah, thank you so much, Parimita ma'am. Once again, a warm good morning to all of you. Progress in science depends on new techniques, innovative discoveries, and creative ideas. Today, AI is turning yesterday's biological impossibilities into today's realities. To truly understand the importance of AI in biological world, it is essential that we come together with a collective vision to discuss and to work towards the solution for the challenges of tomorrow. Hence, the idea of this conference on AI and in silico insights in biology was fostered in our mind, which was uplifted by our management, the Seva Sadan Trust, firmly supported by our principal, strengthened by the encouragement of Microbiology Society India, and shaped into reality by the dedication of the organizing team of this conference. The theme of our conference reflects a transformative phase in scientific research. Artificial intelligence is just not a tool of computer science. It is reshaping the way we understand the biology. From predictive modeling to drug discovery, in silico approaches are shaping innovative solutions with speed and precision. At this moment, I also take the opportunity to sincerely acknowledge and thank all the advisory committee members from India and abroad for constant encouragement and valuable guidance. We are deeply honored to have amongst us eminent dignitaries, scholars, and researchers whose contribution continue to build the future of science. I would like to extend my heartfelt appreciation to my mentor, Dr. Devjani Dasgupta, Professor and Director of School of Biotechnology and Bioinformatics, D.Y. Patil University, for immediately embracing my dream come true idea of connecting with us through this conference and for graciously accepting our invitation to deliver the inaugural address. I have been fortunate to learn many things from her during my doctoral journey, especially through her futuristic ideas that still continue to inspire me. My sincere thanks to Dr. Sarita Mahajani, presently associated with the University of California, USA, for supporting this venture wholeheartedly and for accepting the invitation to deliver her insights on AI. I am equally grateful to Dr. Anil Fokmare, Technical Director at Omnicals Pharma, Portugal, a renowned industrialist in the pharmaceutical sector, who, despite demanding schedule, kindly accepted our invitation. A special note of gratitude to Dr. A.M. Deshmukh, National President of Microbiology Society India, for always standing by us with his strong support and encouragement. I also thank to all our judges, Dr. Manish Kumar from AIIMS, Dr. Rohan Roy, National Institute of Virology, Dr. Dhananjay Mankar from Tata Institute of Social Sciences for readily accepting our invitation. We are also proud to share that prior to this conference, we successfully conducted 1-month certificate course and hands-on workshop on basic bioinformatics and its role in drug discovery, which saw enthusiastic participation from all over the India. Today, most of those participants are extending their learning by presenting their work in this international forum. In this context, I would also like to acknowledge Mr. Nishat Supugade, who recently appointed as Food Safety Officer in Indian Railways, Ms. Malika Ahuja from Taiwan, and Ms. Saili Chabukswar from Italy, who have played pivotal role in training our participant and organizing the workshop, because which has added immense value to this deliberation. This gathering today is a testament to the power of collaboration and shared vision. As Albert Einstein rightly said, the only source of knowledge is experience. And with more than 150 participants joining us from across the globe, spanning undergraduate, postgraduate, research scholars, and faculty members, we are creating an experience that will truly enrich the scientific community. We have shortlisted 35 abstracts covering diverse aspects of AI and in silico research. This presentation, spread across 2 days, will enrich the futuristic knowledge base in biological sciences. On behalf of my entire team, I extend a warm welcome to all the participants, dignitaries. May this conference spark meaningful discussion and offer an insightful journey into the transformation world of AI and in silico field. Together, let us embark on this exciting path of knowledge, innovation, and collaboration. Thank you and wish you a truly enriching and inspiring experience. Over to you, ma'am.
Thank you, Ranjana ma'am, for your insightful words and for beautifully highlighting the theme of this conference. With great pleasure, I now invite our co-convener, Dr. Meena Rao Madam, to introduce our esteemed guest, Dr. Devjani Das Gupta Madam, for the inaugural address. Over to you, ma'am. Over to you, Rao ma'am.
Yes, ma'am. Joy.
Thank you, ma'am. Much. Artificial intelligence is the microscope of digital age, revealing life's pattern in silico. That is the theme of today's conference. Dr. Devjani Gupta is going to give us an insight into this fascinating world. But first, let me introduce this eminent personality who has accepted our invitation and made us a very happy host to have her amidst us. Dr. Devjani Gupta is currently serving as the Director of School of Biotechnology and Bioinformatics at D.Y. Patil, deemed to be University, Navi Mumbai, as a position she has held since 2012. With her illustrious career spanning decades, Dr. Das Gupta has played a transformative role in advancing research, education, and technological innovation in biotechnology, bioinformatics, biomedical engineering, and food sciences. Under her leadership, the School of Biotechnology and Bioinformatics, premier institution earning national recognition. The school was listed among the top 10 promising technology colleges in India in 2025 by Higher Education, along with prestigious institutions such as IIT Kanpur and ICT Mumbai. It has also been featured in the Economic Times as a key player in the sunrise sectors of higher education. The institution has received multiple accolades, including excellence in innovative practice academics at College Dunia Connect 4.0 in 2024 and excellence in biosciences education in 2023 from the Education Minister of Maharashtra. A distinguished researcher, Dr. Das work in anti-cancer drug discovery, bioinformatics has led to numerous high-impact international publications. Her expertise is further demonstrated by her tenure as a postdoctoral fellow at the of Cancers, USA, supported by the Cancers Health Foundation Fellowship. She has served on various prestigious academic and ethical committees, including the Board Committees, Academic Council, and the International Animal Rights Committee. Beyond her administrative and contributions, Dr. Das Gupta has pioneered multiple national and international conferences, fostering global collaboration in biotechnology. Her visionary leadership continues to drive academic excellence, research, innovation, partnerships, making feature in bioscience education. Ma'am, we are really proud and honored to have you with us today. Welcome, ma'am.
A very good morning to everybody, and thank you for this really elaborate introduction. I must congratulate all of you, for this effort that you have put in to light the lamp of, inquisitiveness and inquiry in this beautiful field of in silico biology, which is now powered by AI and has proven to bring in infinite possibilities, right? So, congratulations, all of you, once again for this wonderful effort. I saw the papers that are the nice 50 papers I saw with a large number of participants from across India and abroad, right? So, uh, we have to see that what happened to the IT sector a few decades ago is now happening in the biology sector, right? So it is a kind of a revolution, I must say, in silico biology, and powered by AI has brought in a kind of a revolution in the field of biotechnology and in the field of biology as such. I always, maintained that a good, well-trained biologist will never be out of job, because you have a host of organisms changing their personalities, and the biologist goes chasing those new personalities to understand. We have seen with the COVID virus, and we see with all kinds of microbial organisms. We've seen what kind of antimicrobial resistance is there, and we go after all of this to make the world a better place, to provide health, healing, to provide food, and to bring in sustainability. So this is the job of the biologist, and in silico biology has brought in, lot of insights into actual biology. Now, coming to the topic of this conference, what is in silico biology? So, if you say, it is the use of computational models, computational tools, computational simulations to do experiments, as opposed to what we did earlier, when we were biologists about 40 years ago, when we were doing our PhD. That time things took ever so long, and things were so, expensive. The time and the cost factor has now been drastically reduced with the use of in silico tools. And this has impacted, and this is kind of inclusive of modeling of cells, what all is possible, what kind of experiments are possible by in silico biology. That is modeling of cells, organs, or even entire organisms, simulating biological pathways and disease pathways, predicting molecular interactions, designing drugs computationally. I am told that the other day I was reading a Harvard University newsletter, and I was told that and I read that there are about 10 raised to 60, active pharmacology compounds all designed, with the in silico processes. So that is outnumbering the number of stars in the night sky. So can you imagine what infinite possibilities are possible through this? AI and ML have stepped in to power the biological data analysis sector. A lot can be done in that field. Molecular docking and dynamic simulations are possible. Computational genomics, transcriptomics, proteomics, we are teaching all of that in our department, and any bioinformatics institution would be teaching all of that. And AI based diagnostic predictive modeling in healthcare. Systems biology, network analysis, all this is possible under this experimentation models. Computational approaches in microbiology, virology, structural biology, protein engineering, and digital tools for epidemiology and disease surveillance is also taking place. But most importantly, in our country, for example, if you look, drug discovery and development is highly impacted. There is a lot of deep learning happening there. Reinforcement learning is happening. Generative models are there for you to virtually screen compounds, to create de novo molecules, and predictive pharmacokinetics is possible. The tools such as atomize and in silico medicine, etc. And we are also living in an era of personalized medicine. So, personalized, precision, predictive, preventive medicine, all of this is possible through in silico biology, right? If you look at India, we are also having a bio E3 policy. This bio E3 policy is trying to make India the biomanufacturing hub of the world. Right? We have seen it during COVID times. We have seen how India has provided vaccines, right, for the whole world, and the in silico biology has helped immensely. AI and ML has immensely helped in biomanufacturing practices. And that is going to be the focus of India now. For that, we will need students, we will need lots and lots of young people to come into the sector and be interested, right, into this field. And how do we reduce costs? How do we, make efficiency higher? All this will have to be taken into account. And the Bio E3 policy will really help, the government of India's Bio E3 policy will really help India and the students, faculty, all personnel in this sector to really try their skills out. There is also a speedy growth and evolution of computational approaches in systems biology and quantum computing, right? And a shift towards data driven sciences, right? Which is paving the way for scientific innovation and deep tech startups in India. I just heard a talk the other day which says about 2,000 incubatees happen every year in India over the past few years. And so we are really, having a very good and fertile ecosystem for innovations and in the field of biology, right? Which is there to produce all kinds of products. Biosimilars have come into the markets. Biosimilars will come up, and biopharma is going to rule the roost now, right? So, with all of this, the industry also requires talent. So, my few words would be only that the future will provide you with immense, immense possibilities, right? With in silico biology and with AI and ML tools, there are just immense possibilities that are going to happen. And just to mention a few futuristic things, you will have completely enabled simulated drug development pipelines. You will have driver organ on chip You will have the possibility of digital twin technology, right? So this is something really, fiction coming true now. But we have to focus on our skills, right? The main challenges are market access and skills, right? And so I am going to urge all my young friends here to take their trainings very seriously, to take trainings, as many trainings as is possible, right? And to rise to every occasion. You have to walk the talk, deep sleep, think, breathe biotechnology, breathe in silico biology, and think of different possibilities, right? If you believe me, I have students, 25-year-olds, coming and saying, ma'am, we want to work in the field of in silico biology, and give us projects of that kind. And at the same time, in the same breath, I say that I have, 75 plus year olds who want to, who have retired from their busy schedules, biologists, of course, who now want to come back and do another PhD, a meaningful project, so that they learn the new excited, the excitement, and they are part of the new excitement that is in silico biology and AI and ML possibilities, right? So, students, you're in the right place at the right time. I remain as excited as the young people that I can see here. And I wish the organizers complete success, a grand success for this wonderful conference that you are holding. Thank you very much.
Thank you so much, madam, for your inspiring words. We truly value the way you so eloquently woven biology with AI and in silico approaches, bringing immense depth and significance to our conference. I would like to kindly invite Meena madam to introduce today's distinguished speaker and eminent resource person, Dr. Sarita Mahajani madam, to all the participants. Over to you, Meena ma'am.
Thank you, Parinita ma'am. Dr. Sarita Mahajani is a personality who does not need any introduction. Very close to our hearts, she being the alma mater member of our college, and we being, groomed by her. It is like talking big to the sun. But anyways, the job has been allotted and I will take it seriously. She is an accomplished microbiology teacher with a comprehensive blend of hands-on professional and academic experience. Her expertise in bacteriological techniques, aseptic handling culturing, and characterization through a wide variety of bacterial laboratory research techniques is unparalleled. She has she has experience in running bioassays, laboratory bench studies, molecular and cell biology techniques, maintaining lab operations and equipments, and writing scientific reports. She is trained in fermentation engineering and upstream bioprocessing and have knowledge of current good manufacturing practices. She teaches courses in the Department of Sciences, Mathematics, and Biotechnology at University of California, Berkeley, and also in AI in Pharma and Biotech from Massachusetts Institute of Technology, Sloan. Ma'am, very proud to have you amongst us.
Good morning, distinguished personalities, colleagues, and curious minds. I would like to thank first the organizers for allowing me an opportunity to talk. A special thanks to Dr. Meena for such a nice and inspiring introduction. I am more inspired to achieve more, learn more, get more knowledge. Very many thanks to Dr. Ranjana for inviting me, having a special invitation here. And it's an honor to be with you today at a moment when the boundaries between the disciplines are dissolving, where biologists and computer scientists, microbiologists and data engineers are speaking a common language, the language of data, patterns, and possibilities. So to start with the statement, if the 20th century was a century of DNA, the 21st century will be the century of AI-driven biology. Centuries of human history have been shaped by technological revolutions. And this is going to be a biological revolution, which is coming up in the 20th, 21st century. We are heading towards, as I said, a biological revolution. The advancements made between biology, synthetic biology, are incredible. But what is driving this revolution? In the past 2 decades, we have seen major changes in biology. The genomic revolution is where we learned how to read the genetic information, that is DNA of a cell, at a scale and at a low cost. But why AI in biology now? Just 20 years ago, genome sequencing was a decade-long billion-dollar endeavor. Today, it's a weekend project. The result, a tidal wave of biological data, from genomic sequences to microscopic images, from protein folding data to environmental metagenomes. The challenge is no longer just collecting data. It is understanding it, making sense of this overwhelming complexity, you can say. This is where AI steps in. So artificial intelligence is observing the patterns, principles invisible to the human eye. Biology today generates massive data sets: genomics, transcriptomics, proteomics, metabolomics, biome data. Then you have thousands of microbial communities from gut, soil, water, etc. Then the imaging data from high-resolution microscopy, time-lapse videos. Each dataset is huge and multidimensional, requiring computational tools like AI, ML, bioinformatics to analyze it. So a data model is actually a conceptual representation, you can say, of all the data structures and their relationship within a system. It acts as a blueprint for how data is organized, stored, and accessed, ensuring consistency and efficiency in data management and usage. Going to what Galileo, the father of modern sciences, wrote once, the universe cannot be read until we have learned the language and become familiar with the characters in which it is written. So biology is just not that simple. It's complex, it's messy. If understanding biology is hard, engineering is even more harder. So Python is the most popular machine learning programming language, but a no-no to biologists. Generally, we read 3 simple ideas existing. Everything derives from code. The second, code keeps evolving. And the third idea is we are moving from digital code to reading the live code, to copying the live code, to actually writing live code and thinking about how this stuff is structured and why it's so important that all is derived from code, right? So you see, complexity is inherent to both biology and code. Effective code emerges from simple code interactions. No one actively sets aside to write down a bad code. But good code also does not emerge passively. It delivers from intentional applying to and concentrating on the content of your problem. So put the right tool for the job. That's what they say. We have low code platforms, tools, and platforms that have built in libraries like Jupyter and Colab. And now we come to a new understanding, a new platform, you can say, which is called as no code or zero code tools for non-programmer biologists. It's very inspiring, I can say. That's what I have studied. That's how I am studying artificial intelligence. No code is a method for creating and employing applications that eliminates the need for coding or be familiar with the programming language. For example, we have two beautiful platforms for bioinformatics. One is Galaxy Interactive Platform, open source, web-based, no code, or you can say low code platform for data intensive medical research and bioinformatics. You have Omicshare Tools, a zero code interactive online platform for biology and data analysis and visualization. And then we have the most prettiest one I can see, which I liked very much, was Google Teachable Machines, free online platform by Google, super beginner friendly. You train the models just by uploading the examples. The output is ready. It is ready to use model for ML work and a good platform to study. The next one is Orange Data Mining, visual programming for ML and data analysis. This has inbuilt classification, clustering, visualization all in one and all ready-made platforms. You can cluster antibiotic resistance profiles of clinical isolates very easily in this particular programming, or you can platform, you can say. So these are all the user-friendly platforms. Let's take a quick tour of a few transformative intersections where AI is already redefining or reshaping biology. One is predictive medicine. All models forecast disease risk years before symptoms appear. Personalized treatments, not one size fits all, but one patient, one therapy. Next is the drug and antibiotic discovery. It's a generative AI design. Molecules never see in nature, and it's best for de novo antibiotic discovery and development. So antimicrobial resistance is met with in silico antibiotics tested before they enter a petri dish. Synthetic biology, the hot topic of today. AI will help design more microbes to produce fuels, degrade plastics, manufacture vaccines. Here, biology is used as a programmable system. And the last one, global health and ecology. Air-powered surveillance predicts pathogens emergence, outbreaks from animal reservoirs, and we already have climate microbe interactions modeled to anticipate shifts in agriculture and health. Let's go a little more and see what these things say in the coming few years. So predictive medicine, an area of healthcare that forecasts the likelihood of disease occurrence and helps to implement preventing measures which averts or significantly reduces its adverse effects. It uses a patient's individual data, such as genetic profile, medical history, to forecast the probability of them developing a specific disease. That is called as predictive medicine. It utilizes genetics, biomarkers, imaging, the patient-specific information to enable individual treatment and thereby increasing treatment efficiency as well as lowering the side effects. So the key component is personalized and preventive medicine, which AI is very helpful in because it analyzes the big data too easily. Drug and antibiotic discovery is what you say, what every institute or every research centers are after. Recently, numerous methods have been successfully applied to predict drug response levels precisely. However, most of them target at phenotypic screening, but do not come along with the reasonable interpretability, rendering disease or drug reaction mechanisms very opaque or very obscure. To accelerate precision medicine, it is crucial to explain the mechanism of action of drugs and thereby promote novel drug discovery. Right? We are just speaking about the key trends of 2024 now in this short talk. And these include, given an input list of molecules, the famous AlphaFold 3, which is a protein structure database, generates the joint 3D structure of the molecules. It reveals how they all fit together. It models large biomolecules such as proteins, RNA, DNA, and even the smallest small molecules ligands, which is a huge category consisting of many drugs. Furthermore, AlphaFold 3 can model chemical modifications to these molecules which control the healthy functioning of cells that, when disrupted, can lead to disease. Then we come to again a very new concept, graph neural network. This is to learn the latent features of the drug's molecular graphs and predict the response level. Molecules are represented as graphs, and the graph neural network is trained to predict their properties. You have convoluted neural networks also. These learn the representations of the gene expression data and combine them together to predict again the neural response level. Another important and very interesting concept which is coming up is drug repurposing. Repurposing, where AI algorithms can identify new uses for the existing drugs. It analyzes the patterns in the molecular data. This speeds up drug development process, especially in urgent cases like disease outbreaks. The best example you can take during the COVID-19 pandemic, AI was used to identify potential repurposed drug for treating COVID-19, such as the famous drug that we used, remdesivir. Yes, discovered by screening vast data sets of existing compounds. So AI models analyze drug target interactions, all the networks in it, looks for overlaps between existing drugs and the new disease pathways. Again, repurposing existing drugs is an alluring way. Why? Because it develops new treatments. It relies on medicines that have been studied, have well understood before, has a safety profile, and have gone through all the regulatory approval processes like FDA. It's very easy to use back these drugs again because the data is ready. Let's talk a little more about the famous Nobel Prize winner AlphaFold 3. Protein engineering stands to benefit from AI. One key area of focus have been the de novo designs of proteins created by in novel protein sequences, which predicts the fold into desired shapes and sizes of all the molecules. So DeepMind AlphaFold has solved an age-old challenge that has stumped the field by achieving improved accuracies at modeling protein tertiary structures of all the known proteins simultaneously. The parent company Alphabet restructured its research division to AlphaFold Protein Structure Database. AlphaFold is an AI system, or you can say it's a program developed solely by Google DeepMind. The initial version in 2018, which won an competition award in CASP 13, is a protein structure prediction. It predicts a protein 3D structure form from its amino acid sequences. They use deep neural network to predict the probability. Significant, different, and more advanced version was developed by Google DeepMind and released in 2020 called as AlphaFold 2, predicting protein structure with extremely high accuracy. It is not surprising that Nature Methods identified AlphaFold 2 protein structure prediction as the method of the year. Simultaneously, after that, in 2021, they released an updated version called as AlphaFold Multimer, which predicts structures of proteins complexes with multiple protein chains. Then in 2024 came Google DeepMind and its sister company Isomorphic Labs, which co-developed and released AlphaFold 3, goes beyond just proteins. It can predict the structure and interactions of almost all the life molecule, including proteins, DNA, RNA, and ligands. Broad range of biomolecule complexes, you can say, right? So it is a testament to the power of artificial intelligence. AlphaFold 1 was a solid proof of concept, AlphaFold 2, a major breakthrough, and AlphaFold 3 further broadens the scope to include virtually all biomolecules. Highly accurate and detailed protein structure models help the scientists design new enzymes, understand protein mutations, and identify therapeutic targets for a disease. I think every industry and company is using these. These data structures, or you can say protein structure databases, is free for commercial and non-commercial use and does not require registration to access the structures. It is co-developed by Google DeepMind and European Bioinformatics Institute, EMBL, EBI, and the AlphaFold server, of course, is a separate web service that uses only AlphaFold 3, and it is only available for non-commercial use and requires a simple sign-up. Coming to synthetic biology, the design and engineering of organisms with new purposes, the design and construction of new biological pathway organisms and devices, or to redesign of biologist system by making changes at the DNA level. All this is synthetic biology. AI simulations are very much being developed to predict how altering gene affects mechanisms or metabolic production, allowing scientists to more efficiently design and engineer cells to produce valuable chemicals, biofuels, and biopharmaceuticals. Multiple types of ML programs are well suited for data processing, integration, and saves the industry time and energy, right? So each production is of, you can say, will help develop design microbes to produce fuels, degrade plastics, and manufacture vaccines. AI's future in global health and ecology is promising, but its effectiveness depends on collaboration and responsible development. There are high hopes that artificial intelligence can help tackle some of the world's biggest environmental emergencies. Among other things, the technology is already being used to map destructive dredging of sand or chart emission of methane, a potent greenhouse gas. So AI-powered surveillance predicts pathogens, outbreaks in animal reservoirs, then pathogen surveillance maps. Then you have AI predicting the microbial networks in soil and ecosystem or oceans. Then you have climate microbe interactions modeled to anticipate shifts in agriculture and health. But apart from all these advantages, how is AI problematic for the environment? This is because the proliferating data centers that house high AI servers produce electronic waste, which often contains most toxic substances. It has large consumers of water. They are, you can say, large consumers of water as well as energy, right? Coming to the little bit into the labs where artificial intelligence can be used and still in progress. One is the beautiful digital twin. It's a virtual model simulation of a real world object, process, or system, you can say. It uses real-time data from sensors, instruments, and experiments. It consists of one physical system and one digital, one data connection, that is either lab instruments or sequencing machines, feed real-time data, and then it has to have an AI analyst or analytics like ML models to process the data to predict the outcome and recommend actions. So digital model, which updates continuously, it's like a mirror, the state, the behavior, performance, it predicts everything, and it just informs or displays everything of its physical counterpart. It can be used to test the current situations, predict the outcomes, and optimize the processes without disturbing the real system. So the twin word comes from an idea that the digital model is a duplicate or a twin of the real entity, right? It's not a new concept. NASA was the first one to use this concept during the Apollo program in 1960s, 70s. They created mirrored systems which was used at the ground-based simulation twin to test solutions before sending instructions to the astronauts, right? So it was described as a digital representation of a physical system that stays linked through data and updates. This technology matured in about, say, 2010, and the name was given, and it came into existence, or the concept expanded beyond engineering to biology, medicine, and biotechnology in 2020s. The application now still working. AI is all work in progress, as I said, right? So it's patient specific. The digital twins like organs, microbiome, tumors, fermentation twins for bioreactors, and whole farm digital twins in agriculture. Coming to something that is used in that helps the AI system. It's a biological research which has increasing dependency on computational tools. Yet many AI models remain opaque or misaligned within specific workflows. Here comes the AI co-pilots now being developed by experimental biology designs. These are interactive AI systems that assist scientists with every stage of the research process, right from initial hypothesis generation to the data analysis. So Copilot is trained on extensive literature, optimized for real-time collaboration, enabling the scientist to test reasoning chains, refine the experimental strategies, and also co-developed early stage ideas. Scientists at Stanford University and Princeton University has created an artificial intelligence system that can serve as a co-pilot for CRISPR gene editing experiments, transforming researchers' approach in genetic engineering. This system is called CRISPR GPT. It represents the first comprehensive AI platform consisting of or capable of designing, planning, and analyzing complete gene editing workflow. Published in an open access journal, Nature Biomedical Engineering, on 30th July 2025, the research demonstrates how large language models of AI can be adapted for complex biological applications. We have already a genetic AI, right? You must have heard about it. But it is a more autonomous system designed to handle specific tasks, end-to-end processes, you can say. And it lacks the human's touch to it. It's like an app or a system. While AI Copilot is user-facing, human interface assistant that helps users with tasks, provides insights, boosts the productivity, and often requiring human guidance for complex steps. A great breakthrough. When we speak of human intervention here now, I would like to call a partnership. Rather than replacing, we envision a co-evolution of humans and machines with AI as a thoughtful partner in the workflow. The term Industry 4.0 was coined to be fourth industrial revolution. In biological sciences, it is Bioindustry 4.0 or Biopharma 4.0, referred to as a higher level of automation for operational productivity and efficiency, connecting the virtual and the physical world in an industry. This was, there was no access to, or there was no drive for personalization here. Then came the term Industry 5.0, which was coined to address personalized manufacturing and empowering humans in manufacturing process, and with the key features of having human-machine collaboration. Like robots and AI don't replace humans, but support human creativity and decision-making. For example, in biotech labs, AI suggests the microbial designs or protocols, but scientists guide which one are ethically and practically relevant. So it's a collaboration between the two, you can say. And this pillar of 5.0 is based on three pillars, like 3 points. It's human-centric, it's sustainable and resilient. Systems designed to adapt to crisis, for example, pandemics or supply chain shocks, you can say. And it revolves around these three steps or these three processes. Every terminology or technology comes with, not terminology, sorry. Every technology comes with limitations or challenges. We must be clear-eyed. Bias in data can lead to biased results in AI models. There is another problem which is called as black box model. This can hinder trust in medical decisions, especially. The black box pattern or problem is AI referring to a lack of transparency in advanced machine learning models, like particularly deep learning systems, where it's difficult to understand how they arrive at their outputs or decisions. The lack of interpretability, of course, hinders trust and accountability. So that has to be taken care of. Next comes the most important point, ethical concerns about editing life itself from microbes to humans, and it must be a part of our dialogue from day one. To sum up all, advanced machine learning systems such as deep neural networks are revolutionizing many scientific disciplines as well as our daily lives. The biological sciences have been a fruitful testing ground for the application of these systems. Why? Mainly for two reasons. On one hand, modern biology is characterized by generation of huge voluminous amount of data, which is beneficial for these data hunger systems whose performance depends on availability of a large data, a good data, a structured data, unbiased data to train their sets. On the other hand, the intrinsic complexity of biological systems and only partial understanding of underlying complexity, or you can say underlying mechanisms, the association between inputs and outputs of machine learning system, which learn from training data without awareness of these underlying mechanisms, offers enormous potential for biological systems modeling. That is the key idea why biology is succeeding so much. It has a complex data which has to be analyzed. The objective of this talk was to provide an overview of the current landscaping, providing an insight not only of the state of art, but also of the future potential of these technologies as well. I would like to conclude saying, as data science technologies continue to evolve, we can expect even more transformative applications in the field of microbiology, and this will expand to new generation AI models along with generative AI integration with quantum computing, and will show progress in biomanufacturing, automated scientific discovery, to be in short. Right? So let me now tie everything we have explored into a bouquet. Each flower in this bouquet represents a theme which touched on today: predictive medicine, antibiotic discovery, synthetic biology, ecology, and ethics. Alone, each one is beautiful, but when we bring them together and place an AI in the center as a ribbon that binds them, we see the bouquet. The future biology with AI. It's cohesive, it's connected, and full of life. So can we now say the next era of biology will not be discovered, it will be redesigned. And I would like to stop here saying thank you to each and everyone.
Thank you very much, madam, for your inspiring talk. It was insightful and thought-provoking, enriching our understanding and broadening our horizons. Biologists often feel apprehensive for coding, but you beautifully resolved this by introducing user-friendly platforms and demonstrating their applications. The session highlighted how biology can be effectively integrated with the AI and in silico approaches using user-friendly platforms while providing deep insights that enriched everyone's understanding. On behalf of the organizing committee, I would like to express our sincere gratitude to Mr. Sanjay Dabrai, President of Seva Sadans Trust, our respected principal, our distinguished resource persons, and all the participants. Your support, presence, and contributions have made this inaugural session truly meaningful, providing us all with a wonderful opportunity to learn, share ideas, and grow together. This is an announcement for the participants. After a short break, we will resume with technical session 1 paper presentation at 11:30 a.m. sharp. We warmly request all session chairs, judges, and participants to rejoin promptly, as your timely presence will ensure a smooth flow of the session and allow us to engage fully with the exciting research being presented. Thank you all.
Just a minute, Parimita ma'am. I think there is one question. If Savita madam would
Okay.
like to answer that.
Okay.
Yeah,
Okay, can I read it?
yeah.
Yeah, ma'am, the question one of the participant has asked is, madam, kindly elaborate on how we can make regulation of cell as AI driven.
Regulation of cell as AI driven.
Yeah.
I did not get the question actually, but I can tell you that AI is all about modeling. The models are prepared by feeding in the existing knowledge, data, and they are trained, that's what you say, and then they are put for test and then evaluated. That I know. How to, I did not I don't know. I did not answer that properly, I think. But I don't know how the question is framed. I'll just look it up and come up sometimes. You can mail me.
Yeah, ma'am.
Okay.
You have actually fairly covered on the basis of the thing that we can gather from that question. Thank you so much.
Thank you.
Thank you, everyone.
Also, the announcement regarding feedback. I think Dasgupta ma'am, you are on mute.
Yes, thank you very much. It was a wonderful session. Dr. Mahajani, I just enjoyed your talk. Yeah, all that we do came back. Thank you
you.
so much, ma'am.
Thank you,
Wonderful.
madam.
Wonderful.
My pleasure. Thank you.
All the participants, please fill the feedback forms. Link is given in the chat box. Sir, the link is given in the chat box. Before leaving, all the participants are requested to fill the feedback form.
Good morning, everyone. We are ready to begin now. I will first take this opportunity to introduce today's judges who have made this process of presentation a very easy task and a journey for us. Our first judge for today is Mr. Rohan Raj Roy. He is a passionate PhD scholar with 3 years of dedicated research experience in immunology and virology, specializing in the immune response to Zika virus infection. He is proficient in advanced techniques such as B and T cell epitope prediction, molecular docking, and serological assays, demonstrating a strong foundation in both theoretical knowledge and practical application. He has a proven track record of contributing to peer-reviewed publications and presenting research findings at national conference, showcasing a commitment to advancing scientific understanding. He is passionate about leveraging expertise in immunoinformatics and experimental methodologies to drive innovative research initiatives. His research involves understanding the immune response in Zika virus infection using animal models. He has authored and co-authored 2 research articles in peer-reviewed journals such as Frontiers in Immunology and Journal of Infection and Public Health, respectively. Sir, we are very honored to have you with us. Welcome. Our second judge for today is Dr. Manish Kumar. He is associated with Magad University, Bodh Gaya, India. He is a scientist C, non-medical, under the project Setting Up of a Nationwide Network of Laboratories for Managing Epidemics and National Calamities at State VRDL, Department of Microbiology, All Institute of Medical Science, Devgarh, Jharkhand, India. He is a guest faculty at Department of Botany and Biotechnology, College of Commerce, Arts and Science, Patlaputra, University, Patna. He was awarded Best Poster Presentation Award in 2nd National Seminar on Nanomedicine and Medical Devices in Healthcare, organized by School of Pharmaceuticals Sciences, APJ Stay University. He is skilled in real-time polymerase chain reaction, ELISA, microbial culture, nucleic acid extraction, microscopy, homology modeling, molecular docking, immunopeptide designing, and phylogenetic analysis. He is trained in homology modeling, molecular docking, and phylogenetic analysis bioinformatics in the project entitled In Silico Structure Prediction of Kalpin-like protease of Leishmanian Donovani. Sir has 15 book chapters, 48 publications in international journals, and 26 publications in conference proceedings. His H index is 11 and I-10 index is 13. Sir, blessed to have you with us. Welcome. I call upon now Navya Ramchandran. Before that, if judges would like to address the participants, presentation participants, either of the judges?
Hello.
Yes, sir.
Yeah. So, yeah. So, as this seminar or this workshop is about AI, immunoinformatics, and related approaches like computational studies also. So, all the participants should be like they should be presenting in a lucid manner, and they would have prepared very well and have come to deliver the talks. So, it should be time constrained, like the time limit which is there. You should wind up in that and be ready for the questions. Yeah, I wish you all the best, all the participants. So yeah, let's begin. Yeah, thank you, ma'am.
Thank you, Rohan Raj, sir. Manish, sir? Any
Yes,
word of...
sir. We will proceed. Thank you.
Welcome, sir. Navya Ramchandran, kindly start the presentation. She's the first participant, sir. On your list, she is AI IS5. IS5. She's a UG candidate in silico characterization. Yes, Navya, please start.
A very good morning, everyone. Before I start, ma'am, can you please confirm if my screen is visible?
Yes, it is visible.
Okay, sure. Yeah, thank you so much. So, a very good morning, everyone. I am Navya Ramchandran, coming from PES University, Bangalore, and I will be talking about the in silico characterization of hypothetical protein RV1360 in Mycobacterium tuberculosis, specifically the H37RV strain. So I'll start off with a quick introduction, talk about the scope of my research, a methodology, and then move on with my results, finally with inferences and my references. So, a quick introduction first. So, Mycobacterium tuberculosis is a bacterium that causes tuberculosis, as the name suggests, but it enters the human body through aerosols and it settles in the human lungs, where, when it is phagocytosed by alveolar macrophages, as you can see, they tend to live on the nutrition or nutrients provided by the cell, and then they form granuloma, where they exist as latent TB. But when this granuloma ruptures is when it goes into active circulation and it forms active TB, affecting multiple other organisms, not just the human lungs. So I took this project specifically because there is increased drug resistance in Mycobacterium tuberculosis, and that leads to a reduction in treatment efficacy. One, two, current treatments include long treatment periods and very strong antibiotics, and also inconsistent antibiotic dosage or consumption by patients worsens the issue. So we need to find newer drug targets that can actually present to a viable solution. So this is my methodology. I started off with protein sequence acquisition from UniProt, and then moved on with homology search using BLAST-P, and multiple sequence alignment using Clustal Omega. Of course, moving on with analyzing protein parameters with tools like ProtParam, Interpro, and gene ontology databases. And finally, the conserved domain database search, homology modeling, ligand search, and finally, understanding the active sites and the amino acid biochemistry present there. So, starting off with the conserved domain database, I found out that RV1360 is a probable F420 dependent oxidoreductase, meaning it uses coenzyme F420, and it's pretty similar in domains to bacterial luciferase. And finally, it belongs to the superfamily of flavin utilizing monooxygenases family. So this coenzyme F420 is actually a flavin type of compound. So if you look at this multiple sequence alignment, starting off with the phylogenetic tree, it's a very basic one where I started off comparing RV1360 with F420 dependent oxidoreductases. And then it is one of the Mycobacterium tuberculosis oxidoreductases with a percentage identity of about 99.71%. And then further computing protein parameters using ProtParam, I found out that the whole protein had around 340 amino acids with a molecular weight of 37 kilodaltons and 6.38 theoretical pi. And it classified that the protein was relatively stable, and a negative Gravy index or grand average of hydropathicity shows that it is a hydrophilic protein. Using Swiss model, I got the homology model, and as you can see on the right, there is the Ramchandran plot showing that most of these amino acids are present in the dark green region where there are most favorable dihedral angles. And using PyMol for visualization, I found out the active site. And as you can see here in yellow, that is a ligand I found using the chemical EMBL database with a lot of surrounding amino acids. Further visualization using LoicPlot and BioVia Discovery Studio revealed that there were two main hydrogen bonds with glutamate 223 at one position and methionine 174 at another position. And as you can see, it is surrounded by a lot of hydrophobic interactions as well. And so finally, what I concluded from all of my research was that it is a probable oxidoreductase, number one. It is relatively abundant, that is around 33.7 parts per million abundance. And that was taken from PAXDB, another protein database, which uses flavin as a substrate rather than a cofactor. Usually molecules like FMNH2, that's reduced flavin mononucleotide, is generally used as a cofactor to provide electrons or protons as required by the enzyme. Over here, on the other hand, coenzyme F420 is used as a substrate. So it catalyzes the synthesis of virulence factors, I mean this specific protein, or any proteins that use a coenzyme catalyzes the synthesis of virulence factors like PDIMs, and also the detoxification of antimicrobials. It can confer resistance to drugs like delaminid and pretominid, both of them which block the production of mycolic acids, enabling for increased survival rates of this bacterium. So coenzyme F420 in general, it protects against oxidative stress like a reactive oxygen species or reactive nitrogen species as well, and promotes the persistence of the pathogen in the host, and it acts as a cofactor for enzymes like F420 dependent glucose 6-phosphate dehydrogenase. We'd wonder why this is relevant to RV1360, right? Apparently, this enzyme, glucose 6-phosphate dehydrogenase, reduces F420, coenzyme F420, adding a hydrogen group, which further is used up by the Mycobacterium tuberculosis or the bacteria or the pathogen to activate these drugs. When the drug is ingested by the host, the reduced coenzyme F420 dependent enzymes tend to activate them. So that might be a potential function for RV1360. And what makes and the cherry on top is that it is specific to Mycobacterium tuberculosis and making it an effective drug target because it doesn't attack or influence any of the other host proteins, reducing the amount of side effects. So these are my references. Thank you.
Very well done, Navya. Dr. Manish, Mr. Roy.
I am Manish here. Dr. Navya, I am very happy being a UG student. You are presenting such a nice presentation. Uh, I will one question from my side that how many types of docking you could be performed apart from this for the
So I have
project?
actually done blind docking only so far. I haven't learned any other types. So using that as a tool, I moved on with the active site detection.
Okay. Can you use server or software?
I used PyRX software.
Pyrex. You
Yes.
also go with many software servers that perform docking. It will give a uniform data analysis. Not first, as a student, you can go for the blind docking. It's good, but when you proceed learn more things, you put to the focus to the main size. Okay.
Okay,
Thank
yes.
you. Thank you.
Thank you so much, sir.
Manish,
Yeah,
sir.
so Rohan, hello. Hello.
Yes.
Yeah. So Navya, like you presented really well. As sir also told, like a UG student, but you presented in a very lucid manner. So can you go to your slide which shows the epitope, the H bonding interactions, hydrogen bonding interactions?
Yes, sir. Yeah, I hope this is the one, sir.
Yeah, this one. Yeah.
Yeah.
So like, how did you perform it? Like you told like you did blind docking, right?
Yes.
So then how did you come out with this, the interactions and all? In which software did you do this?
So first, I did blind docking using PyRX, and
Yeah.
then used BioWire Discovery Studio, as you can see in the
Yes.
left, to visualize these interactions. And on the right is using LigPlot Plus.
Yes, yes, yeah. So that I wanted to know. Yeah, this is through a Discovery Studio, and the right side is of Liquid.
Yes.
So, like you told about the in silico analysis and all. So, if as a like you'll be doing research in future, or you'll be going for masters and all. So, like, how can you deal with this problem, keeping the in silico part aside? Like you have explored the AI and in silico part. So, any other ways, the future prospects you would like to say?
So I'd like to clarify. So are you talking specifically about docking or
Wet lab, I'm asking. You have
wet lab?
told about, yeah.
Okay, so I'm not exactly sure, but I would maybe try to use some sort of spectrophotometry, maybe cryo-electron microscopy, one. Sorry, not spectrophotometry, pardon me. Cryo-electron microscopy or X-ray diffraction, or even NMR to actually look at the interaction, so the dynamic nature of the protein and ligand. I'm not sure if that's possible though, sir. So please forgive me if I'm wrong.
No, it's fine. Like the things you told are quite similar on the lines. Yeah. So like it was a good presentation. Thanks a lot. Thank you, ma'am.
Thank you, sir.
Thank you, Navya. You can stop sharing. Yeah. Next, I call upon Arnab Rishika Kole, PG category. Rishika, start please. Your slide is visible, Rishika.
Okay, ma'am. My name is Rishika Kole. So today my topic is AI integrated hydrogel based strain sensor as wearable device in health monitoring application. Healthcare today is changing very quickly. People want devices that are not only accurate but also soft, comfortable, and smart enough to monitor health in real time. Traditional sensors are often hard, uncomfortable, and not fully suitable for our bodies. To solve this problem, we have developed hydrogel based strain sensors combined with AI. The need of such devices is growing because of chronic disease, the demand for preventive care, and the importance of remote health monitoring. Today, I will be explaining why AI integrated hydrogel based strain sensors wearables are required and how they can improve the future of healthcare for everyone. So, smart wearable are and stretchable strain sensors are widely explored for health monitoring, motion detection, as they can detect little changes in the body and convert them into electrical signals. Conventional materials like metals, graphene, and carbon nanotubes have been used, but they lack stretchability, stability, adhesion, and self-healing, limiting their use in long-term wearable devices. So, hydrogen, on the other hand, are soft, flexible, biocompatible, and mechanically similar to human skin, making them excellent candidates for strain sensors. So wearable health monitoring devices are portable, sensor-based gadgets that can be comfortable worn on the body. Some common examples include smartwatches, fitness bands, and biosensor patches. These devices are biosensor are designed to track vital signs such as heart rate, blood pressure, oxygen level. The biggest advantage of these devices is that they provide real-time health tracking, which helps in the early detection of the potential health issues. At the same time, they support personalized healthcare and play important role in managing chronic diseases. So, importance of real-time non-invasive monitoring. First, it helps in early disease detection and prevention, since even small changes in the vital signs can be captured immediately. It also leads to enhanced patient engagement because people can actively track their own health. For those with chronic disease, these devices support better long-term management by providing continuous updates. AI can analyze individual data and give tailored health insights. Real-time monitoring also enables proactive interventions, allowing doctors and patients to act before condition worsens. This is only reduces hospitalization and healthcare cost, but also improves recovery and smoothens the transition from hospital to home. So, strain sensors in health monitoring devices are soft and flexible sensors that can be worn on the body. They detect small changes in shape or movement and turn them into electrical signals. This allows us to monitor things like body movements, posture, pulse, and even vibration of the vocal cords without any invasive movements. They work on a simple principle. When a force is applied, the sensor stretches or bends, and its size or shape changes slightly. These changes affect its electrical property like resistance or conductivity. The sensor then converts these changes into signal that can be read and analyzed by electronic systems such as Arduino. So this is a working of a strain sensors. A strain sensor checks how much something bends or stretches or compresses. It does this by measuring resistance in thin wire or foil. If the wire or foil is stretched, resistance increase. If it is compressed, resistance decrease. A metal foil pattern is fixed on a flexible surface. This foil acts like a measuring part. A small current flows through the foil. Suppose the object bends, the foil also bends. Because of this bending, stretch foils gives higher resistance and compressed foils gives lower resistance. The resistance changes is very small, so we need a smart way to detect it. That's where the Wheatstone bridge circuit comes in. It has 4 resistors connected in a diamond shape. 3 of them have known values. One is the strain sensors, which gives unknown value. When the resistance changes in the strain sensors, the bridge gets unbalanced. This unbalance produces a small voltage difference as the output. That voltage directly represents how much the material is stretched or compressed. So this is a hydrogel. A hydrogel is a network of cross-linked polymer chains that can absorb and retain large amount of water or biological fluids, resembling a natural tissue. These soft, flexible materials can be triggered to change shape or properties by stimuli like temperature, pH, or light, making them suitable in diverse applications such as drug delivery, tissue engineering. So the main characteristics of hydrogels are they are water absorptive, have cross-link network, they are soft and flexible, and also they can be used in various biomedical applications. So this is a hydrogen-based strain sensors. Hydrogen-based strain sensors are smart hydrogen with properties like self-filling, stretchability, and strain sensitivity are very promising for wearable devices and electronic skin. Among natural polymers, Tara gum is useful because it is biocompatible, holds water well, and forms flexible gels. Its chemical structure allow easy modification, giving the hydrogel strength, electricity, and ionic conductivity. Ionic hydrogens are important in strain sensors because they convert body movement or stress into electrical signals. Tara gum hydrogels can be made using borax, which forms reversible bonds and also helps in
Rishika,
ionic
your time
conductivity.
is up. Please conclude.
So these are the hydrogen materials that can be used for strain sensors. One is synthetic hydrogens and natural hydrogens. This is a research gap in which we have included AI over here. This is a synthesis process of the polyamine hydrogen. So this is a result in which it's showing that the hydrogen having more porous structures which helps in ionic conductivity. This is a TJ analysis which shows that it is temperature stability, and the glycerol that has been mixed in the hydrogel showing anti-freezing property in the hydrogel. This is an antimicrobial property where A. aureus and E. coli is against of antimicrobial property. Means the hydrogel are against of this A. aureus and E. coli. This is a self-filling property where the hydrogel can be self-filled, and it also acting as a strain sensing property when the hydrogel bends according to the position, and also this and also having addition property also. This is the electrochemical performance of this is the electrochemical performance of the TG hydrogel. To test research, we have connected to a circuit that measures resistance while a finger bends and stretches. The hydrogel stretches, its internal network is disturbed and resistance increases. So this is a real-time response of the hydrogel. The system works like the hydrogel is connected to an Arduino Uno microcontroller, which measures changes in resistance. These analog signals are sent via serial communication and displayed on a Python interface as percentage changes in resistance. The setup allows us to continuously observe dynamic human motions in real time, demonstrating the sensor's practical potential for wearable applications. So these are the experiments that we have conducted.
Rishika, I'm sorry to interrupt you, but you should go to the conclusion now.
Okay, ma'am. So this is the application base where we can use these hydrogel based such as in hand tremors, joint movements, and early signs of the mobility issues like in Parkinson disease. It is also helpful. Advantages: it is highly flexibility, biocompatibility, and AI powered. Challenges such as it has continuous sensing plus AI processing drain energy, need for efficient low power, long-term stability is not there, signal noise is problem. And the conclusion is hydrogel-based strain sensors are flexible biocompatibility. Integration of AI enhances accuracy. Together they enable personalized healthcare, and also AI is there. This is a future direction where development of low-power AI algorithms, integration of the energy harvestation such as motion, heat, solar, enhanced stability, and advanced AI integration should be also there, and improved variety should be also be there. These are the references. Thank you.
Judges.
Yeah, so Rishika, like I would like to give some suggestions in the initial part. Like when you are telling about AI and like related informatics and all, but most of your topic was kind of a wet lab kind of thing, right? And also don't put so many texts in the slides. Minimize in the future, like you are students, right? So in the future, keep the punchlines rather than going for all full texts and all. Okay. And one question which I have for you is, so how do you how do you know about the gate analysis? Can you tell GAIT, the gate analysis? How can it be implemented in this monitoring the health monitoring and hydrogel based strains sensor?
So that kind of software will be implemented in future.
No. Like I'm asking, like if you no, that is already been done. So I'm asking, like how can you put your topic on, like move it to this part, like the gate analysis? Like they are what? They are wearable hydrogel sensors integrated into the knee braces, right? So how do you go about it? What is gate? Can you say GAIT?
Sir, that is the this analyzes the posture of the movement of uh, of a person who is suffering from neurodegenerative diseases such as
Yes.
Parkinson or Alzheimer.
Yeah, so that was my question. Like, if they are like people who are rehabilitating or people who are, if you monitor certain athletes, the movement and locomotion, right? It comes into play. So, like you could have told like how it your topic can be integrated into that. Yeah, overall it's fine. But next time when you go for future presentations, keep your slides to minimum, and also punchline should be there rather than so many texts. Yeah, thank you, Risika.
Dr. Manish.
I am agree with Rohan that there are too much text in the presentation. I have only one question that what advantages, one or two advantages of doing using hydrosol materials that provides for strain sensors in comparison to the traditional electronic components?
Mm. Sir, the hydrogen is biocompatible, and also it's suited for human skin,
Okay.
and it also provides instant drug delivery when it will require.
Oh, thank you.
Thank you, Rishika. Now I call upon Mr. Arnab Jana. Please join. Stop presenting, Rishika. Mr. Arnab Jana. Your slide is visible.
Good afternoon, ma'am. I am Arnav Jana. I am from Laminar University, Department of Allied Health Science, Bachelor of Science in Medical Laboratory Technology.
Arnab,
Okay,
kindly put, hello, Arnab, put your slide into presentation mode, please.
Yes, ma'am.
Presentation mode, beta. Sit in presentation mode.
Slide show karo, slide show karo.
Yes, ma'am.
Yeah, thank you, beta.
Yes, ma'am. Today my topic is artificial intelligence for
Sorry,
clinical flow
Arnab.
cytometry.
Arnab, you have joined with two devices. That's why echo is there.
Mute one of the devices, please.
Okay, ma'am. Today my presentation topic is the AI automated flow cytometry. Nowadays, AI is an essential diagnostic tool for the hematological and immunological malignancy, cancer, and any other autoimmune diseases in the clinical laboratory settings and other researches. In this study, we was the creation of fluorescence activated cell sorter workflow. This helps to detect the hematological malignancy like acute myeloid leukemia, acute lymphoblastic leukemia, chronic myeloid leukemia, lymphoma, and multiple myeloma, etc. through its automated photo detector, and it is helps to reduce the cases in 5 minutes. So I am going to discuss about the facts. Means fluorescence activated cell sorter. It is a specialized type of flow cytometry. It is advanced versions of the flow cytometry. Here we can sort a heterogeneous mixture of biological cells into two or more continuous at the one time at a one time based upon their clinical light scattering and the fluorescence characteristic. It is a flow cytometry. It is a technique which helps to determine the morphological structure like cell size, cell shape, the chemical characteristic like internal complexity and the granularity of the cells. In this process, the sample is injected into the narrow chamber and the laser beam. So the laser beam shows a cells in scatter. It is characteristic. When it was forward scatter, it determines the cell and size of the cell, and when it is the side scatter, it determines the internal complexity and granularity of the cells. Now I am going to discuss about the three components of the facts. First, fluidic system. Second, optic system, and third is the electronics part. Now I am going to discuss about the flow system, fluidic systems. In the fluidic systems, the main principle of the fluidic system is the hydrodynamic focusing. Without any physical barrier, two fluids, seed fluids and biological body fluids are passes in the narrow chamber, and then uh, uh, narrow chamber and without any barrier, uh, and it is passes through the narrow chamber in front of the laser beam and detect in the photo detector. The optical system consists of sensor, various filters, AI photo detectors, and the dichroic mirror, which is situated at the 90-degree angle. Here you can see the optical system beside these pictures. The cell, when the cell are flows to the narrow chamber, to the in front of the light, it excrete different types of wavelengths structures. Then I am going to discuss about the electronic system. These band single monochromatic light band plus signals are converted into the electronics system data, which helps to us data visualization, interpretation with the help of hybrid network. And in this process, not only complete blood counts like RBC count, WBC count, and their subtypes, we can determine the platelets and different types of hematological cancers and autoimmune disease like CD19 plus and CD20 plus autoimmune disease can like systematic lupus erythematosus, CD plus help for the HIV positive patient, CD for CD16 plus helps to the malignancy and cancer, CD30 plus for aplastic anemia, CD180 plus for the multiple myeloma, CD130 plus for acute myeloid leukemia and acute lymphoblastic leukemia, CD125 for ovarian cancer, and CD25 for hairy cell leukemia. Then I am going to discuss about the fluorescence activated cell sorter applications. Uh, with the help of with the help of the forward scattering, we can detect the morphological structure of the cell size, cell particles, and the surface area, different types of side markers and CD markers. And the side scattering, we can detect the granularity, internal complexity of the cell, like specific antigens, cytokines, B cells, T cells, like helper T cell, cytotoxic T cell, natural killer cell, and many more. And enzyme activity we can also detect. Not only lab detection, it also helps the biomedical research like immunology, cancer research, and the stem cell research. In case of immunology, it can detect the specific types of lymphocyte cells and each subtypes like T cells, B cells, and natural killer cells. With the help of fluorochrome surface markers like CD plus surface marker, it also helps to detect the cancer uh, like multiple myeloma, lymphoma, chronic myeloid leukemia, and acute myeloid leukemia.
Arnab,
It all
your 6 minutes are up.
yes,
Wind up,
here
please.
are the reference. Thank you, ma'am.
Very well done. Judges?
Yeah, Hello, Arnab. So you presented about the flow cytometry AI application. So how like most of the slides you told, they were about the principles, the basic understanding of flow cytometry and all. So are you aware of immunology, like viral immunology, because this topic is related to that? Arnab?
Sir, there was the significant development of the AI photo detector, which helps to detect the
No, I am asking, are you clear about some immunological concepts? I will ask a question regarding that, related to your flow cytometry. So the question is like, you told about many cell types like CD4, CD8, B cell, NK cell, and all. So
yes,
just a basic principle, how will you differentiate between CD4 and CD8 in case of flow cytometry?
in case of flow cytometry, we can divide the CD4 and CD8 different types of anti HIV antibody or anti HIV and their surface area.
Are you done?
Different types photo detector AI
First of all,
with the
just
help
calm
of
down a
photo
bit.
detector we can differentiate the
First,
CD8 and
calm
CD4.
down a bit, and just relax and say you are taking too much stress, so that your voice is also not clear. So the basic is like CD4, CD8 will have anti CD4 antibodies will be there, markers will be there. They will differentiate rather than going for HIV, because HIV is a disease. It's any disease through the markers we can differentiate, right?
Yes,
So
sir.
that should be your answer. Okay. And one more question is like, you know about, like, if there is any disease outbreak or outbreak of any disease, you want to know about predict clinical outcomes, then how will you use AI in flow cytometry to predict clinical outcomes?
With the help of AI, as the AI software is fully automated, so the clinical diagnosis and analysis are reduced to less than 5 minutes. So we can determine these types
Okay,
of
thank you.
malignancy
Thank you.
disease and hematological pathogens.
Okay, thank you.
Thank you.
Dr. Manish?
Can you go through the main technical or operational challenges in integrating AI to in flow cytometry? Hello,
Hello.
hello, Arnav.
Hello, sir.
Can you go through our main technical or operational challenges in integrating AI tools in flow cytometry in patient scenario?
Yes, sir. AI flow cytometry is very useful for detect the different types of disease very quickly. So it is very useful and
Oh,
necessary.
okay, okay.
Thank you, sir. Thank you, Arnab.
Thank you.
Next participant, Mr. Ravikant Masi, please join. Your screen is visible. Put into slideshow. Yes. Ravikant, please start. Judges, Ravikant is a PG student.
Say,
Ravikant, please start.
Am I audible, ma'am?
You are now audible, but your screen is freeze.
Okay, okay, ma'am. Is it now like
This is your first slide?
yeah, this is my first slide, and this will be the short
Yes,
introduction
yes,
kind
yes.
of.
Please start. Please start.
So the very basics of the introduction of the how AI is integrated into the ECG. So here what I can show is you that what ECG does is it takes the data of the ECG. The AI takes the data of the ECG, those PQRST complex, and then it kind of analyzes it accordingly. There are some of the models which uses based on the AI, which will analyzes the ECG, and based on that, what we can figure out is future arterial fibrillation, coronary artery disease, cardiomyopathy, and some AI-guided management can also be done in acute coronary syndrome, pulmonary embolism, and heart failure. So, how does AI does it is through pattern recognition. So, through the lens of pattern recognition, AI unveils the subtlest harmonies with the EKG, unraveling mysteries that have long eluded the human eyes. Human interpretation, though skillful, can sometimes miss the finer nuances that the trained AI system can discern. So here, the little bit of background is that EKG is a key tool for non-invasive cardiac diagnostics, and manual interpretation is kind of time-consuming and complex. So AI in its deep learning model is enable automated with a high precision diagnostics. So pattern recognition is where the AI capabilities truly shine. The AI algorithm is trained on extensive data sets of EKG, exposed to countless instances of normal and abnormal heart activity. So through this training, the algorithm learns to identify the intricate patterns and relationships that a human might overlook or struggle or to recognize. So the aim of the presentation is the main goal of the presentation is to understand how the AI enhances the ECG analysis. So there are three key aspects how it does it. So arrhythmia detection, the first one, the prediction of the structural cardiac abnormalities, and long-term cardiac monitoring for continuous patient care. So moving on to next slide, AI and ECG data analysis. So here I can say is that AI learns the two types of patterns in spatial patterns, that is the voltage across the lead. So we use in the ECG we use 12 leads. So in those 12 leads, the voltage differences it measures that, and through temporal patterns recognition, the changes in those voltage over the period of time, it learns uh, how to recognize, through that it learns how to recognize some cardiac events. So how it does it? It outperforms the traditional method of speed analysis and accuracy. So there is like a special pattern. As I would like to describe something about it. So ECG leads capture electrical activity from different angles of the heart. AI analyzes the voltage reading and across multiple leads to understand the spatial distribution of cardiac electrical activity. And this helps detect the abnormalities related to heart structure and localized issue like ischemia in specific regions. So this is the spatial pattern. And in temporal pattern, how it takes on the temporal pattern, the variation of the voltage over time reflects the heart rhythm and conduction system behavior. So AI tracks these changes in the waveform, like the waveform of the P wave, QRS complex, and T wave to identify the arrhythmias and conduction blocks or irregular heartbeats. So there are some common AI models. So in the very first slide, I showed that there are some AI models which, in the basic AI models which we use, the social media models, the Grok, this and that. So for this also, there are models which are developed for in neural network in deep learning. So there are three models: convolutional neural network, and Bayesian neural network, and recurrent neural network. So what convolutional neural network is going to do is it is ideal for extracting complex spatial and temporal features from the ECG data. And this model is the newest and most refined and most accurate and most precise network. So this is the one model which I'm going to discuss further. And I'll tell something about Bayesian and the recurrent neural networks also. So Bayesian neural network is useful in scenarios where the model needs to handle uncertain or incomplete data. And in recurrent neural network, especially long short-term memory, these models are designed to analyze sequence of the data, making them perfect for continuous ECG data analysis over a period of time. So that's the difference between these models, and basically focusing on the convolutional, that it takes the data from spatial and temporal feature extraction. So what are the applications? So there are abnormal beat detection, arrhythmia detection, structural heart disease prediction, and risk prediction. So recognizing in abnormal beat detection, recognizing the ectopic or irregular heartbeats. In arrhythmia detection, classifying various types of irregular rhythms like atrial fibrillation, AFIB, or
Shrikant,
ventricular
kindly go
tachycardia,
to
AFC.
conclusion, please.
Yes, ma'am. So this is a short some sort of a flow chart where the ECG data collection, data processing, feature extraction, and data splitting, how these models are getting trained over a period of time, and that's how they take the data. So about the CNN model, this is a very highly accurate model for atrial fibrillation detection, and there are AI accordingly outperforms the expert cardiologist in diagnostic accuracy. And structural abnormalities detection like left ventricular hypertrophy, asymptomatic ventricular dysfunction, heart failure, and cardiac amyloidosis. These are the diseases. These are the events which we can identify. So a little bit of this introduction about these events, and there are long cardiac monitoring, like a Holter monitor analysis. So this is the Holter monitor analysis. This also uses the AI. This is continuous monitoring, which we can get it from the market. And there are some insertable cardiac monitors. So here is you can insert it in the skin, and it can give data accordingly to the doctor as well and to the patients as well on the mobile phone itself. So there is also risk prediction. It can predict the future cardiac events, specify high risk patient, personalized patient data on ECG. So what are the challenges? The challenges are here, and the research gap here is despite its advantage, the AI faces challenges, high computational requirement. There is a high computational requirement in particularly this field because we are dealing with live samples and patients. So AI, it's somewhat in the gray area to be trusted or to be not trusted, but definitely it is going to be helpful. And having a conclusion based on both the doctor's advice and the AI, and so we can use it definitely, but still it needs some of the computational things. And there are data privacy concerns, and there is some black box problem. Black box problem is something like AI is not supposed to be biased, but somehow it is getting biased and uh, giving advices which are not supposed to be given. And there is limited generability due to biased data sets. Overcoming these requires continuous research and collaboration. So ethical consideration, what we are going to see is that responsibility is misdiagnosis. The ethical issues include the responsibility for incorrect diagnosis and clinician trust in the AI. To address these things, what we need is that AI literacy programs for healthcare workers, development of explainable AI models, and this all although this needs a large amount of funds and government approvals, but there is a long way to go there. And so future direction is that integration of multiple models, the one which I told you about, the CNN, BNN, and the RNN. So what we can do is integrate it, but still there is a need of human intervention, and it is cost effective, and also ongoing research is overcome existing limitations. So conclusion, AI models revolutionize the ECG interpretation by automating detection and prediction of the heart condition with high accuracy and speed. And it enables personalized patient care, reduces diagnostic workload, and assists in early detection of the risk. And however, the collaboration between data scientists and clinicians is essential for effective implementation. So that's the end of my slide.
Yeah, thank you so much. Kindly switch on your video, judges. Ravikant, kindly switch on your video.
Uh,
Can stop presenting and switch on the video. Participants, during the question answer session, switch on your videos, please. Manish sir, Rohan sir.
Ravikant, I have one query, that what are the potential benefits? Okay, what are potential benefits and limitations of using AI in ECG detection or analysis?
The potential benefit is that as a human we have limitations. Uh, the very short, the very subtle things like interpreting the T PQ or T interval, and sometimes we kind of miss it as a as a doctors kind of miss it. So analyzing the huge data sets and collecting it and then interpreting it, it is not human. It is beyond human capability, and that's the point it has. But the limitation is same like the points which are favorable in the AI. The limitations is that sometimes it misses some of those things, and kind of biasness it get that is the black box theory. So there is some kind of biasness in the AI, and they are trying to figure it out. How does it uh, how does it predict, and uh, does it think? But what we need is the human intervention, and the training has to be given to the doctors as well to analyze with AI and give a conclusive report to the patient. It can be very fast, but we need to analyze. Yes.
One more, can you take concern of those patients who are using pacemaker?
Yeah, we can, but as I told you, it's a very gray area. You see, these models, the CNN, the BNN, the RN, they are still under research, and uh, we we pacemakers are fine with it, and uh, but the problem with that is you have to be very careful accordingly. So. Uh,
Rohan sir.
Hello, Ravikant. Yeah, the topic which you presented, it's like really a good one. Like because AI, most of the people think that AI cannot be integrated much into the this ECG and all. But you have ticked some boxes with like through the computational neural networks and all.
Yes.
So, can you differentiate between CNN, a computational neural network, and random forest algorithm? What is the difference? Like you would have known about this random forest algorithms also, Bayesian, random forest, etc.
Yeah, Bayesian random. So the difference is that recognition of the CNN is through spatial patterns and temporal patterns. So what it does is that it captures the electrical activity and voltage differences. And in case of Bayesian, it is what it does is it kind of continuously tracks the things like scenarios where the model needs to handle uncertain or incomplete data. Some data is incomplete and it tries to complete it. That is Bayesian. So it predicts it and
And like
completes it.
about
Though it
C,
doesn't have a data, but it completes it. So, and in recurrent
so
time,
what about CNN?
so CNN is the extracting for the complex spatial and temporal data. So spatial and temporal, I've already explained it that electrical activity across different voltage parameters, and temporal is that the changes for a longer period of time. So that CNN does, and it is the latest one, and it is far more with 95% around accuracy, but it's still we need some lot of people computation.
Okay, so one more question is, how does it promote the personalized cardiac care? Like, how are you looking in that aspect?
I think
Because you told about the gray areas and about the black box and all. So how does it, if you go for personalized cardiac care, that is a quite specific term, right? So how do you go about it?
so, what I think is maybe in developing drugs in pharmaceutical things we can personalize uh, for a for a patient, and that we can do, and personalized care can be given for that.
Oh, oh, thank you.
Welcome. Well done, Ravikant. You can leave now.
Thank you.
Next is Miss Munalika Mahakur. She is a UG presenter, sir. Stop presenting, Ravikant.
Yes.
Munalika, kindly present. Your screen is visible. Put into slideshow.
Good afternoon, everyone. So I am Munalika Mahathur. So today I will be presenting or speaking rather on the topic emerging digital and analytical tools in disease detection.
Munalika, your screen is not visible yet. Munalika, your screen is not visible yet.
So I will be speaking on the topic emerging digital and analytical tools in disease detection, surveillance, and epidemiology. So first for the introduction, I would say that in recent years there has been a significant shift in the fields of modern medicine and epidemiology. So the
Pardon me,
advent of
Munalika.
digital innovations and artificial intelligence, big data analytics, and mobile health
Munalika,
technologies.
Munalika, your screen is still not visible. Let it be in that mode only. Don't go to the slideshow. Share again.
Am I audible?
You're audible. Now the screen is also visible. Let it be this way. Continue, please.
Madam, kindly request her to switch on the camera.
Yeah, Munalika, kindly switch on your camera.
Hello?
Munalika, yeah, switch on your camera, beta. Yes. Now continue, please.
Am I audible?
Yes, yes. You are audible. Monalika, you are audible. Monalika, you are audible to us. Monalika, till you sort this out, we will move on to the next candidate. Is that okay? Monalika, till you sort this out.
I'm having a problem. I will fix it and then I can present.
Yes, please.
Okay, thank you.
Yeah. Preetham, kindly join and present.
Yes,
Preetham
ma'am.
Das.
Yes,
Yeah.
ma'am. Yeah, join.
Judges, this is a UG candidate.
Ma'am, am I audible?
Yes, yes, you are.
Okay, ma'am.
You can present, switch on your camera too.
Okay, ma'am.
Share your screen, please.
Okay. Ma'am, is it visible?
Yes, it is visible. Put into slideshow mode.
Now is it visible?
Yes, yes, yes. Please go ahead.
Good afternoon, everyone. My name is Preetham Das, and I'm currently pursuing Bachelor's in Medical Lab Technology in Brainware University. Today, I want to speak on the topic accurate point of care powered by AI-based biosensors. So let us go from the basics. What are biosensors? Biosensors are the devices that detect specific analytes using recognition elements, converts the uh, interest reactions into signals, and via transducers the signals are processed and the data is interpreted. So the aim of this review is to see how AI integration helps in point of care biosensors. Integrating AI with point of care biosensors aim to uh, create more intelligent, rapid, and personalized diagnostic systems leveraging AI for the data processing in real-time analysis. Moreover, AI enables the real-time data analysis, adaptive learning, and predictive results, which improve accuracy, efficiency, and early disease detection. Ultimately, this means better patient care and greater accessibility in healthcare settings. So actually, how AI enhances the biosensors? AI enhances every stage of the biosensors development, from selecting the right analytes to designing the recognition elements, improving transduction, and refining data handling. The goal is not only accuracy and speed, uh, but also to personalization and usability in real world conditions. Now let us look at the market trends of the biosensors. Currently, the biosensor market is one of the fastest growing segments in the healthcare technology. Uh, Rising demands of point of care diagnostics, patient and doctors are increasingly prefer quick and accurate tests outside traditional labs, especially after COVID-19 pandemic. Integration of wearables devices like smartwatches, fitness trackers, and even the patches are now embedding biosensors, and enabling it with the AI gives the continuous health update in real time. Let us see the process by which the AI can enhance the biosensors. AI can choose the analyte selection. It also helps in recognition of elements and also the transduction of signals. And at last, it also helps in interpreting the data handling through the help of machine learning and deep learning codings. So one of the important topics is wearable point of care with help of biosensors. Let's go through some examples. For instance, the GX sweat patch developed by Epicor Biosystem in collaboration with Gatorade, this microfluidic device analyzes sweat in real time and uses smartphone images processing to provide insights into hydration and electrolyte balance. It is a clear example how AI turning raw biosensor data into personalized feedback. Moreover, the urine and saliva-based biosensors are also there. Diagnostics is pioneering in saliva-based biodiagnostics to detect multiple diseases. Wearable diapers, which have built-in glucose measuring uh, capabilities, helps to detect various diseases non-invasively. Moreover, the Neuralink is also developing some technology where we can just control our machines through just thoughts only, like telepathy. So why should we integrate AI with point of care wearable biosensors? Because it enables the continuous monitoring of the biomarker uh, and also facilitates personalized treatment in specific and shows specific insights. It also helps in early detection of diseases like epilepsy and seizures. And also, by integrating the human reasoning into explainable AI models, we reduce the amount of time uh, and training data required. Leading companies are already investing in heavily in this field. Like Abbott Laboratories are expanding continuous glucose monitoring system in AI integration. Roche is developing AI-powered diagnostic tools for faster point of care. Medtronic is pioneering an AI-driven diabetes management solution, and Dexcom is advancing continuous in glucose monitoring with AI analytics. Uh, this shows that how AI integrated biosensors are not just future, but also the present, rapidly growing in
Preetham,
adoption.
kindly go to conclusion, please.
Okay, ma'am. To conclude, the integration of biosensors, AI, and microfluidic technologies present a gradual shift in healthcare. Wearable biosensors provide continuous, non-invasive monitoring, and artificial intelligence interpret these vast data sets and deliver accurate, real-time clinical insight. Together, they are transforming diagnostic from episodic testing of continuous, predictive, and patient-centered care. Ultimately, the synergy will pave the way uh, of healthcare that is not only precise but also accessible and quotable. Thank you very much for your attention.
Judges.
So, Preetham, it was a good presentation. You stuck to the time, and also your slides were like not too much elaborated, it was precise. So, if you want to integrate AI into these biosensors, okay, so what will be your future strategy regarding this AI in biosensors? Like whatever has been done till now, what you would look to find something novel in it?
Sir, first of all, I would try to develop the same technology which are present here now. After developing those, I will try to find the answers of some questions, like how it is done or why it is done. And after finding out the answers, I will try to integrate um, our machine learning or deep learning or some neural networks to the biosensors, which are till now not available. Means I will try to do so, but after learning through different types of um programs and other, I will try for doing it.
Yeah. So you'll go about the whys and hows of the topic. Yeah, that's a good way to approach a question, a research question. Yeah. Thank you.
Thank you, sir.
Very well done, Preetham. You can.
Hello, Pritam, one question from my side.
Yes, sir.
What are the technical problems which need to be solved to connect AI algorithm with the biosensors or
Um,
biosensor devices?
Sir, first problem is that providing the data set and also the information to the AI, how we are going to interpret the result. One of the problem is that. So after resolving that, we can go further in launching the product and also using it for the further uses.
Oh, and data collection?
Yes, sir.
Can AI tool help in collecting data? through biosensor? It could be.
Yes, it could be possible.
Oh, thank you. Thank you.
Yes, sir. Thank you, Preetham. Ranit, kindly join. Start presenting. Yeah, your slides are visible now. Put into presentation mode, slideshow. Switch on your video too. Ranit, switch on your video too.
Yes, ma'am. I have switched on the video as
well. Yeah. Please continue.
Good afternoon, everyone, all the dignitaries and the judges present here. I am Ronit Majumdar and I am from Benway University. So my today's topic is that it is a study for AI, that intelligent portable laminar airflow. So we have developed a portable laminar airflow, the conventional ones which are very heavy and expensive, and which is not convenient to be transported from one place to another. So we have tried to develop one miniature version of a conventional laminar airflow, and within that we have incorporated various sterilization uh, uh, items as well, and we have integrated the AI version in this, and which allows us incorporating various sensors, and which we can use to monitor the growth of the microorganisms, and which can also give us various informations about when actually it is the best time for the growth and what temperature it is required for that. So our aim was to actually develop a lightweight and a portable laminar system, and which gives us the utility to use aseptic conditions, the sterilizations, and with a very less interactions within the convenient places. So generally, we have developed it in a very miniature form and used very basic items which are readily available. And we have used 360 plastic, which is very readily available and very cost effective. And along with that, we have used battery powered LEDs, and the stands which we have made that are of wooden type. So, and we have incorporated various sensors to detect the temperature, the humidity as well. And also we have given motion detector sensors, so that the UV light which we use actually sometimes while operating in the laboratory, we tend to forget the switching off the UV light, and which can cause many mishaps. And we have used monitoring device which senses the motions of the hands, and it automatically switches off the UV lights. And we are also doing modifications that which so that we can incorporate our Wi-Fi system. Along with that, we can generate. We are further working on this process, that we can develop a mobile application through which we can monitor each and every utility of the every aspect of the growth of the microorganism. So the AI incorporations which we have done actually, which uses the temperature and humidity, that DHT11, we have used that sensor, which actually senses the which actually works upon 3 volt to 5 volt within the voltage range, and it can also detect 0 to 50 degree of temperature, Celsius of temperature as well. So here is a video of that. So where we can detect the motion. So when the motion is there, when the lid is open, the light automatically floats up, and when the lid is closed, it switches off the light. And the hardware which we have incorporated, that is Arduino Uno R3 sensors, which actually transmit the signals into a data, and we have used an OLED display so that we can see the temperature which is within the laminar flow. And we have done various field surveys from within our college and within our across various hospitals which are present within our locality. So we have found that within the conventional one, they face a various problems, and which we have developed a portable one. So they find it's very easily available. It can be easily available if we further proceed. And we have also done the patent. We have done the patent work, and it is at the ideal stage, and we are waiting for the grant. And the commercialization can be done further up. So what we actually provide, the uniqueness within ourselves, which actually we provide a compact laminar airflow system, which is very durable. This is a diagrammatic sketch of our portable laminar airflow, and which we actually have made a prototype, which I have seen mentioned earlier. This is a picture of our prototype, within which we have given an the pink color box, which is being seen. That is actually the sterilization one, because we actually in the laboratory we actually sterilize ourselves, but when we actually work with the microorganisms, we can also again it can be helpful for our more sterilization process, disinfection process, sorry. And the uniqueness which actually a sensitive automatic sanitizing system, which I have already told, and
Ramesh,
constant. Yes, ma'am.
kindly go to conclusion now.
Yes, ma'am. So we have also studied the market, and we have generalized this. And in conclusion, I would like to conclude that which we are providing, that which we have made, it also provides a less expensive one than the conventional one, and it can be readily available at the field, where we can do various cultures if we need a instant basis. If a samples is ready, and we need to collect it on spot and have to culture it. So the portable one will make it easier to do. And in the rural areas where such conventional laminar airflows are not available within the laboratory system within the hospitals, so we can provide this miniature version with a less expensive one, and which will be energy efficient. So with that, I would like to conclude my. Thank you.
Judges.
Yeah, so Ranit, like I would really appreciate your like work, because you have at such an young age you have gone about of modifying or preparing a laminar airflow with such modifications in a resource limited setting. So that's a really good job. And so my question is, like, I was really happy to know when you told about the UV will go off after some
Yes,
time,
ma'am.
right? After
Yes.
how much time it goes off when you
If we
forget
actually,
to switch
it
it
is
off?
basically a motion detecting sensor we have put. So if we remove the hand, it automatically, if we insert the hand within the lid, so it automatically switches off. And if we remove our hand, it automatically switches
Okay.
on. And if we cut off the power supply, it automatically put down everything.
So when you remove the hand and you go away for other work, so it will automatically
Yes,
switch it off, right?
switch on.
Yeah.
And if we power off the main supply, then automatically full laminar airflow will shut down. So that
No,
time
no,
we have,
I'm saying if you are going out, like you are doing some work and you want to
yes,
put the UV on
yes,
and then go do another work and then come back, so that that much time the UV should be off.
yes. So it is done that actually that way. If we remove our hand and we put off the lid, and when we put off the lid, automatically senses that, and automatically the UV light gets on.
Okay, okay, that's really good. And at what stage of patenting are you? Like provisional patent or
Yes, sir, it is an original patent, and we have just
PCT?
done, yes, PCT, and we are waiting for our grant so that we can commercialize it. Next year.
Yeah. So have you like it has been compared with different kinds of like biosafety cabinets, if you see, like these are laminar airflows. If you go for
Yes, sir.
biosafety cabinets, so have you
Yes, sir.
compared the efficiency of both? Like yours is the initial stage,
Yes,
but
yeah,
like
we have made a prototype actually.
yeah.
So with that, we have
So
done our surveys, and
okay,
with that, we got a very good response. And
okay,
everyone, we have collected various reports from the in our locality, within the hospitals, and within various colleges. Because in educational level also, it is very helpful, because they also require in the colleges, so they can easily access our this portable miniature type, so that each and every colleges can also afford it.
yes, yeah. Thank you.
Manish.
Congratulations, you have nice presentation and your concept is very new. Uh, how could you use technology in HEPA and ultra filter?
So actually, so we are
How you incorporate?
we have trying to incorporate it in a horizontal form within our which the prototype which we have made. We have tried to incorporate within that a mesh-like net initially now because we have not done the commercialization. So just for a prototype, we have made that one. But the HEPA filter, which is in general use, we are trying to incorporate that only within our portable client, just in a miniature version.
Thank you. Congratulations.
Thank you, sir.
Yes. Very well done, Rani. Thank you so much.
Thank you, ma'am.
Monalika, you can join now.
Yes, ma'am. So good morning, good afternoon, everyone. I am Nalika Mathur. So today I will speak on the topic emerging digital tools in disease surveillance, detection, and epidemiology.
Monalika, the
As
screen is
in
still the same. Excuse me. We can't see your screen yet, beta.
now,
We are getting a blank screen. Yeah, now we can see the screen. Do not put it into presentation mode because I think there the problem is occurring. So show as it is. Go back.
Okay. Ma'am, now is it visible?
No. Don't go to slide. Yeah, now. Yes, keep it that way.
Yeah. Okay. So the topic on which I will be speaking today is that emerging digital and analytical tools in disease detection, surveillance, and epidemiology. So in recent years, we have seen that modern medicine and epidemiology have seen a significant shift in the fields of disease detection and epidemiology. So the advent of digital innovations, artificial intelligence, big data analytics, and mobile health technologies is revolutionizing the methods that were used to detect, monitor, and manage diseases. So, coming to the modern diagnostic systems, in the modern diagnostic systems have begun to incorporate both computational and biomedical technologies. For example, if it's about the artificial intelligence and machine learning, it has enhanced cancer detection up to about 94% accuracy has increased. And devices like smartwatches and glucose monitors also are used nowadays. So coming about the smartwatches, now some smartwatches can detect the arrhythmias like atrial fibrillation, etc., using their integrated electrocardiogram system or different sensors. And in the case of glucose monitors also, we have seen that using a sensor and enzyme, it creates an electrical current so that it is proportional to the glucose that is present in the sample. Now, health map is also one web system that has been now being used to, to do the real-time monitoring and all. So now coming to big data and cloud computing also has helped. And so the impact of this has been that it has helped us to detect various diseases in various ways. So this picture shows that how AI/ML can help in the healthcare systems or how artificial intelligence has helped in the public health system. Like it can help us with the prediction of the diseases or the diagnosing of the diseases, and it can also use a surveillance system so that how much the disease is spreading or not. Now, coming to the digital surveillance, as we know, when mass gatherings occur, there is a huge chance of spreading of diseases and pandemic situations. Like as we saw in the case of COVID-19, also the same thing happened. Uh, that is why we need the social distancing. So when there is mass gatherings, there is a chance of spreading of diseases. So there are types of systems have been implemented, like I have shown here, like the syndromic surveillance systems, the event-based surveillance systems, etc. Here I have mentioned about the web-based platforms like the health maps and ProMed. These are basically the web-based platforms that they have created a data and they have seen how the disease has spread and have incorporated in that way. So the 8% of the studies has
Nalika, it seems you have network problem. You can switch off your video and continue.
Okay,
Let's see.
okay. Yes.
Continue.
Yes, ma'am. So coming to the digital surveillance, etc., as I was speaking about. Now coming to the different tools that we have.
Okay, it seems her internet problem increased. We'll give her a time period of 1 minute. If she can join, otherwise we'll go to the next participant. Next participant is Tanveer. Kindly be ready. Within a minute, we'll tell you.
Ma'am?
Yes, Monalika, please continue.
Yes, ma'am. So as I was speaking, then I would go to the next slide. That is the mobile technologies during the COVID-19 epidemiology. During the COVID-19, as we saw that there was this outbreak of the disease, and it was very much problematic and hazardous to us that the disease was spreading in the rapid way. So different mobile technologies were used during this time. That is the COVID symptom study app was used for collecting the self-reported data from the different users, and this was the real-time scalable, and it was very low cost also. That is a public health surveillance that was taken in action. Now next I would say that so this is the picture that shows the different digital technologies that were supporting during the COVID-19. Now, that's I would like to say that modern digital epidemiology advances such as remote sensing and GPS nowadays can track vaccination coverage also and human mobility. So mathematical modeling also can help to stimulate interventions for diseases like malaria, TB, and different HIV virus, etc. Reverse vaccinology using genomics sequencing and AI is even helping design the what I would say the vaccines of what people need to take in the future. So with this, I would like to conclude by saying that the integration of AI, IoT, and big data, genomics, etc., and mobile health applications also is reshaping the future of public health. Basically, these tools can help us to allow detect diseases earlier, and to predict the outbreak of the diseases and respond more effectively. So, however, for global impact, if we need to make this happen in a global manner, we must address the challenges. There are different challenges which we might face, such as the infrastructure gaps are there, the ethical governance and different things are there. But with the efforts, we can try to put this into global impact, and with the help of the sustainable development goal also there is there good health and well-being. So with that, we can ensure that there is a healthier and more resilient future. Thank you.
Yes. Ishta?
Monalika,
Yes, sir.
your presentation is well, but so much clumsy. Try to put as less content as possible. Your overall presentation is well. I have one question that how can digital epidemiology tool will help to predict or prevent the future outbreaks?
Sir, we can, with the help of AI, we can make apps that can, do a survey around people, that people can do it like the symptoms as well, or with the help of, the data collection, with the news reports, etc. So we can know that what can happen actually. So with that, the help of that, we can take prevention according to it.
Okay, thank you.
Rohan sir?
No questions, it's fine.
Yeah, Monalika, thank you so much. Very well done. Tanveer, kindly start presenting. Monalika, you can stop.
Yes.
Tanveer?
Yes, ma'am.
Yes.
Very good afternoon to everyone. My name is Tanveer Afrin.
Tanveer, you have to
I
start
am a
presentation,
student
no?
of Masters in Medical Lab. Yes, of course. I have
Tanveer,
to
we
start
are, yeah.
presentation.
Yeah, your slides are visible. Go to slideshow. Go to the top, there slideshow will be there. See above that line, enable editing. Above there, slideshow is there. Yeah. Start now, please.
A very good afternoon to everyone. My name is Tanveer Afrin. I'm a student of Masters in Medical Laboratory Technology.
Also switch on your video, please. Can we switch on the video, please?
Yes, ma'am. Yes, ma'am. I'm visible.
No, we can't see you yet. Oh, not yet. Your video is still not seen. Is anyone able to see?
No, it's blank.
Yeah. She has switched on, but not visible.
Yeah, is your front cam? Your front cam is on.
Yes, Angad. Tanveer?
Yes, ma'am.
Try to switch off and on. Now you can start. Show the video again. Start on the video again. She has started the video, but somehow it's not seen.
So are you doing it on the laptop or on your phone?
Yes, I'm doing on a laptop only.
So
Okay,
is
but
there a cover or something on that laptop camera?
No, I don't think so.
okay, we can let continue with the audio because
Yeah,
it's
yeah. Continue with her.
Okay. I'm starting again, okay? So today I will be presenting how AI is revolutionizing breast cancers under the topic AI and in silico insight into the biology. So first, a breast cancer and overview. What is breast cancer? So a breast cancer is a disease in which abnormal breast cell grows out of control, proliferates and form tumors, and it can occur in part of the breast tissues, male ducts or ropules. There are about over 2 million women were get diagnosed in 2023 and are predicted to increase 70% in the next 20 years. So the early detection of breast cancer is substantial to fight against this disease. And now here where AI is emerging as a transformative local, offering more accurate and more personalized insight into the cancer management. So you can see here there is a two images in which one anatomy of normal breast anatomy and second one is the cancerous breast anatomy. So what are the major challenges are facing by the women in breast cancer? So mammography limitation. Poor mammography images are not effectively visualized tumor in women and affect the accuracy of breast cancer, increasing anxiety, stress, and deploying overall health. Now, second point is false positive and false negative. Screening method can produce false result indicating cancers when it is not there. Or false negative result missing an actual cancer delayed treatment. Variability radiologist interpretation. Different radiologists may interpret the same mammography images differently. So the breast cancer diagnosis can be hampered. And then the dense breast tissue. Dense can cause overlapping of tissue because some men have more fatty position on their breast, so reducing the visibility of cancer images.
Tanveer, we can't hear you.
Uh, 1 second, ma'am. I'm moving to the next slide. So, how role of AI in breast cancer detections, early detection? Now moving to the AI in breast cancer detection. Analyzing medical images mean interpret mammograms, ultrasounds, MRI, and DBT scans to detect signs of cancer, such as abnormal growth tissue changes and calcification. Highlight the area of concern. You can see the images, how AI highlight the area of concerns. Improve detection rate. AI-assisted screening has increase in cancer detection rates compared to traditional method. Okay. AI can detect breast cancer up to 5 years before diagnosis uh, by visualizing risk factor of the woman. Enhance medical workflow. By working with radiologists, AI can act as a second reader, speed up the reading process by pre-screening large volume of mammograms and uh, act to assess cancers that might be missed by human review alone, thus reducing workloads of radiologists. Personalized risk assessment and monitoring. AI can generate risk score to estimate an individual likelihood of developing breast cancer based on their unique risk factors.
You must go to conclusion now.
So how AI algorithm? Should I understand this slide?
Yes, please, but go to conclusion then.
So how AI algorithm works? Okay, algorithm has a set of instruction. Okay, first the key product is data collection. Means AI algorithm of we have to fed the algorithm with large amount of data to learn the
Tanveer,
various
please go to
pattern.
the conclusion. It's like your slides are taking too long to be communicated. Can you please go to conclusion?
So I'm going to the conclusion that AI can improve breast cancer detections and diagnosis accuracy. AI-powered predictive models can identify high-risk pattern of predict treatment outcomes. Personalized treatment developed with AI can improve treatment efficiency and reduces side effect.
Finished?
Yes, ma'am.
Yes. Judges?
So Tanveer, it's your topic. There is a word called theranostics, right? In the initial, in
Yes, sir.
the title of. So what do you mean by theranostics?
Theranostics. Theranostics is a combination of therapy and diagnostic into a single.
Okay. It was not audible. Like, can you again say?
Theranostics is a combination of two words, therapy and diagnostic, that combine into a single word, theranostics.
Oh, okay. Yeah.
Manish sir?
Ranjit, one question only.
Yes, sir.
What challenges existed in integrating AI technology in curing breast cancer or detecting breast cancer?
So can you please repeat your question?
What are the existent challenges in curing or analysis the breast cancer?
What are the challenges?
Integration of in terms of integrating AI tool.
Sorry, sir, I'm not getting your question.
The challenges existing in integrating AI tool
Yeah.
during or during or in existing breast cancer diagnosis.
The actually there is no simple answer for your question, but I try to give the answer. AI models need to be seamlessly integrated into clinical workflow. Mean they have to update their version again and again and again, so to improve their data and improve this performance.
Okay.
Yes. I have one more question for Tanveer. Like, how do you incorporate multi-omics and radiomics into this breast cancer? Because it is concerned with radiation therapies and all. So how
Radiation
do
therapy.
Yeah. So how do you combine multi-omics
Yes.
and radiomics to treat breast cancer?
So this is a part of AI, but I don't have so much idea about AI. So multiomics and radiomics is I'm not clear about these two terms. Sorry.
Yeah. So you can go about it, read about it a bit. Like multi-omics is a basic part of AI.
Multiomics, yes, sir.
Yeah.
I have
So
seen this word, but
yeah.
unfortunately I missed.
Yeah, you can just go and do some reference, refer back to some concepts. Yeah.
Okay, sir.
Thank you, Tanveer.
Thank you, Tanvi. Very well done. Participants, we will be taking a short break now. We will be starting the next technical session at 2 p.m. sharp. The list is already posted on the group. Kindly go through and accordingly be present. Do leave now, but join at 2 p.m. Rather 1:50 p.m. so that we can start the sessions on time. Thank you all. Judges, kindly stay back for a minute. Participants can leave, can join at 1:50 after the lunch. All the participants can take a break and be back at 1:50, a break of 30 minutes and be back at 1:30. For all the participants, take a break, be back at 2 p.m., 1:50 p.m. for technical session at 2. Patil sir, can you remove the remaining participants for the time being?
Yes, ma'am. May I end meeting?
I wanted to talk to the judges for 5 minutes, then we can end.
Okay.
Is it possible to remove the participants for some time?
Yes. Give me a minute, ma'am.
They have not understood or they have forgotten that they are still here.
Yes, ma'am. Ma'am. Rao, ma'am.
Yes, Patil sir.
Yeah, now judges and we organizers here only.
Yeah. Judges, we will be requiring from each category the best paper for today. Ranjana ma'am, have you joined? Maybe
Madam,
she's still.
Yeah, ma'am, I'm there.
Yeah.
Joined, yes.
Ranjana ma'am will have to get the names at the end, no?
Yes,
So,
it's from each category.
When I was talking to Nishad, I thought there is some confusion. We should clarify to the judges how many names and how many categories they are going to give us names.
Okay, no, today at least from each category we want one name.
I think it is clear to judges then, one name from each category we want.
Yeah, ma'am, it's fine. Sure.
Yeah, and we'll be announcing at the end.
Yes.
Yeah, so I